Hi, everyone. I'm Keoni. I've worked on a few things in the past. Um, I started out at UCI, where I worked 4 years in Cheng Lu's lab on directed evolution and mitochondrial engineering. Uh, Previously, I've worked at Free Genes for 3 years, leading that project in Gerundi's lab at the BioBricks Foundation, where we built cool things like a recoded set of all the JCVI minimal genes and Golden Gate vectors for E. coli expression. More recently, I had co-founded a company called Trilo Bio, building full automation machines for synthetic biology. So not just moving liquids, but low-cost machines to do tube opening and closing, and moving plates in and out of incubators, and doing pipetting. And then during that time, I had built or helped build this open source package called Poly, which is a bioinformatics library for Go. And so all of my bioinformatic tools are kind of built from the ground up, with me really touching every layer. So I'm building a 60 codon E. coli, or 60 codon JCVI-Syn3A. Sorry about the 61. I forgot about the tryptophan that was removed in the code when I went to grab the screenshot. And so I'm recoding all of the TAG codons to TAA, all the tryptophan TGG codons to TGA, all the serine AGT and AGC codons as well, kind of replicating what had been done in E. coli before, but in JCVI-Syn3A. And the hope is that we can kind of make it more routine of building these minimized codon sets for this awesome genome. So why build a JCVI-Syn3? The main reason for me is I want synthesis and construction to be routine and not really special. I think synthetic genomes should be available for less than $25,000, something that you can just buy rather than having to, build out a lab in order to build these genomes. And because there's lots of economies of scale you can get with different ways of building DNA. And then the reason, scientifically unnatural amino acid incorporation, but really I'm just, I want to build it. And so one of the biggest things I think that is preventing things from getting better in the field is that plasmid synthesis costs have actually gone up over time. They've not gone down the last 5 years. Back in 2018, for a 1 kilobase pair of cloned plasmid from Twist, it cost $90. And now it costs about $125. And so even adjusting for inflation, the cost of building plasmids commercially has been going up, not down. And I think that's a real big problem. And as it turns out, oligo pools are much cheaper. If you don't know, oligo pools are in one tube about 12,000 different oligos of 100 to 300 base pairs of length that are all synthesized at once on chips. And so if we can take the output or the input of oligo pools and use that rather than DNA fragments, we can get the cost down of raw synthesis by a huge amount. And so here on the left-hand side is some spreadsheet I've been making, looking at the costs of the per kilobase pair if you're synthesizing DNA with an oligo pool. And on the right is kind of the curves of how much the pool costs up front and then the price per kilobase pair. And so we can see there's some nice logarithmic curves, log curves. As you synthesize more, it gets much cheaper. And so what I do is I clone oligo pools. Here are two of my robots, one doing nanopore sequencing and one doing some PCR prep. The idea is that if I can clone DNA from the oligo pools very cheaply, I can build DNA very cheaply, which makes my plasmids cheap, which makes my genomes cheap. And so this is what I do. The oligo pools assemble just fine. I have evidence of that. It's taken months to really nail it on how to build all these oligo pools in bulk to actually get the genes, but I'm just about there. The costs internally are about $1,100 for me to do 384, and I'm testing techniques to get that down to less than 384 per 384. Right now it costs 1,100 per 384. And so the bulk costs right now are about 1.5 cents per base pair for me to synthesize DNA, but I'm hoping to get that cost down to about 50 cents per base pair, or 0.5 cent per base pair. Sorry. Why does it cost so little? This is just kind of a laundry list. Sorry for all the text on screen. I'm able to get the cost down of building plasmids to this very cheap kind of $384 price by using, I use PCR to index 384 colonies at once for putting things into nanopore preps. Everything's on 384 well plates. Everything's automated on Opentrons. I express and purify my own enzymes. I'm testing different natural competence systems using recursive BSA1. The software is built from the ground up in order to be low cost, to facilitate low cost experimentation and low cost DNA build. I'm working on microchips with some collaborators to do DNA synthesis in-house. I'm working on the first 384 plasmids for this project right now. And so it really comes down to an execution of how low can I drive the synthesis price, so that how low can I drive the assembly costs in order to build these genomes. And so I'm right now synthesizing this 60 codon JCVI-Syn3A. I've synthesized all of the DNA. I just need to start doing the assembly process. It takes about 1,800, 300 base pair plasmids, which then I construct in sequential steps up to 10, 55 kilobase per plasmids, at which time I'd send to our wonderful collaborators at the JCVI to put into yeast and then hopefully do a genome transplantation. And so the hope is to make this kind of thing more routine, so that if we want to make a 55 codon JCVI-Syn3A or less, it will be very easy to do, and we can do that pretty quickly. And so thanks. This is my lab, which is actually in my bedroom right now. Hopefully move out soon, but it enables the cost to be really, really cheap. Yeah, thanks.
Keoni, so when Twist did the Syn3A genes, there were a bunch they couldn't make.
Yes. Twist has about a 20% failure rate.
Mm-hmm.
I'm hoping that I can actually just figure that out, because I'm doing everything in-house and kind of continuously. I'll just retry all the genes. You know, the synthesis price gets down pretty low. The other thing is that the process in which I use does not use Twist's kind of overlap PCR, and so the overlaps can cause, you know, some shifts, or if you have homologous regions, they can kind of flop around, homologously recombine. I'm using a more Golden Gate method, and that's been shown to have much more reliable results, although it requires that you remove BSA1.
Okay. Well, and so as you continue listening today, you will hear Klara Kluchova from the Charles University in Prague, and she is interested in building a minimal cell. She's going to talk about this, a minimal cell that uses fewer than 20 proteins. So there could be some synergy between what you want to do and what she wants to do, that you might get in touch with her after the meeting.
Yes.
Other questions for Keoni?
I saw Daichi's.
There's Klara, or I saw her for an instant. I guess as people make a tiny bit of noise, sometimes we hear them. They show up.
So he asks, why did you choose to remove 2 codons for serine, leaving the other codons? I think mainly on the suggestion from John Glass for serines. The other reason is, personally, is that the serine AGT codon, or tRNA, that John Glass had recommended actually has a BSA1 cut site in it. And since all of my process requires BSA1, if you remove that and, ironically enough, the tryptophan anticodon, you can build a JCVI with no BSA1 cut sites, which enables you to, my system, to work to build the full genome rather than just parts of it.
And I'll add in that when we were thinking about minimizing the minimal cell, we were working with Yale tRNA expert Dieter Saul, and our first thoughts, so he took into account wobble and all of this and all of the things that tRNA people know, and I clearly didn't at the time. And this is his thinking, and this is also the same approach that George Church and Jason Chen used to build their coli strains with fewer than 64 codons. Okay, other questions? I see that, I hadn't been paying attention to chat, but there's an active chat thing going on. So good, but you can also speak like humans. We use our mouths.
I have one question if we have enough time.
Yeah,
this is Sebastian Rodrigue. Nice presentation, Keoni. I was just curious. It's very spectacular to see how you can drive the cost down. But I was curious to hear about the error rate. So you push the cost down, but do you get 100% accuracy when you build these constructs, or do you have to screen a lot of clones until you find one that is perfect?
Yeah, good question. So generally what people try to do, and this is one of the reasons that DNA costs a lot, is synthesize oligos that are higher fidelity, more efficient, longer, like with enzymatic, and then assemble them really well. I'm taking kind of the opposite approach of I use really bad oligos, but I assemble very small chunks at first. And so the target is like 300 base pairs rather than 3 kilobase pairs right out of the oligo pool, so I have to screen less. Now, even though I'm screening less, from the cheapest oligo pools, the mutation rate comes down to about 1 in 392. And I'm building 300 base pair chunks. It comes out to about a 60% success rate per colony picked. And I pick 4 colonies each and then do retries on the next round of cloning. So statistically, it comes out that I can, like, the prices that I'm quoting are all building that in, as well as an 80% success rate, just given randomness.
Can I just ask a quick question?
Go ahead, Bogomil. We have time.
Yeah. So you're building them small fragments, but I am kind of curious about the next steps. How are you putting things together, and where, like, at which point are you putting them to different hosts? Because we know, for example, that this low GC DNA cannot be maintained in E. coli. You mentioned Vibrio. So I just try to picture how we're going to assemble that.
Yeah. So from 300 base pairs, each step of assembly just uses BSA1 Golden Gate. I have a recursive kind of method that uses methylation in order to do that. But the general concept is that I'll clone 300 base pairs, and then those 300 base pairs, I'll clone it to 1,200 base pairs, 1,200 into 5,000, 5,000 into 10,000, and then those 10,000 into a back of about 55 kilobase pairs. And those will have to be assembled together in a yeast cell to get the final transformation. But hopefully we can get about 55 kilobase pairs of the JCVI-Syn3 genome in backs in E. coli.
Before you go to, okay. So you're completely going to be skipping. Okay, that's what I was thinking you would be doing, but just want to confirm. Thank you.